today we are here understanding the new chapter. It is the chapter number 3 of your biology, AS and A level science biology 9700 is the code. Okay. So the first question is that is the enzyme chapter similar to the enzymes the chapter of the O levels biology? So no, although it is a bit same, but there are some changes such as we'll be understanding in this live session that what are the basically new changes, and as you know that they are the practicals in your A levels, so you are also required to do this in your AS level. Okay, so what are the enzymes? So enzymes are the biological catalyst. The biological because they function in living system, and catalyst because they speed up the rate of chemical reaction without being used or changed. This we have learned a lot of time in our chemistry and biology, but what is the something new in your A levels? So is that the enzymes are the globular proteins. Okay, so the critical to the enzymes function is the active site where the substrate binds. Okay, the metabolic pathways are controlled by enzymes in a biochemical cassette of reactions. Virtually every metabolic reaction within living organisms is catalyzed by an enzyme. Enzymes are therefore essential for life to exist. All right. So, what do you think about the enzymes, intracellular and extracellular enzymes? Do you know about it? So the words have been telling that extracellular or intra means something like inside the cell. Yes, intracellular enzymes are produced and function inside the cell, while the extracellular enzymes are secreted by cells and catalyze reactions outside the cell, such as the digestive enzymes in the gut. We'll be taking an example of it as there's a table in front of you. Okay, so the type of enzymes, the examples are given to you, and the function of the particular enzymes is being given to you. So the example of the intracellular enzymes is catalase. So the catalase, first of all, learning the function of the enzyme catalase. Hydrogen peroxide is produced as a byproduct of many metabolic reactions. It is harmful to cell. Catalase converts hydrogen peroxide into water and oxygen, preventing any damage to cells or tissues. So this is the basically function of the catalase. Okay, and what about the amylase, which is an extracellular enzyme? Digestion is usually carried out by extracellular enzymes. This is because the macromolecules being digested are too large to enter the cell. Amylase is involved in carbohydrate digestion. It hydrolyzes starch into simple sugars, or maltose. It is secreted by the salivary glands and the pancreas for digestion of starch in the mouth and small intestine, respectively. Alright, so what is the mode of enzyme action? Enzymes have an active site where specific substrates bind, forming an enzyme substrate complex. What is this? We'll be seeing the figure to understand about it. The active site of an enzyme has a specific shape to fit a specific substrate. Okay, extremes of heat or pH or the concentration of hydrogen can change the shape of the active site, preventing substrate binding. This is called denaturation. All right, that what does it means? That if the pH is changed or the temperature is changed, the enzyme will be denatured. Substrates collide with the enzyme's active site, and this must happen at the correct orientation and speed in order for a reaction to occur. As now you can see this example that where is the active site? Active site is the region in the enzyme where the substrate binds, and then the products are being formed. Before the production of the formation of the product, there is the substrate for a while attached to the enzyme, and for a temporarily structure called enzyme substrate complex formed. This is temporary, after which the reaction starts and enzymes either make the product, okay, from the substrate. Okay, The specificity of an enzyme is a result of complementary nature between the shape of the active site on the enzyme and its substrates. The shape of the active site and therefore the specificity of the enzyme is determined by the complex tertiary structure of the protein that makes up the enzymes. Okay, proteins are formed from chains of amino acids held together by peptide bonds. This is what you need to learn. The order of amino acids determine the shape of an enzyme. All right, and if the order is altered, the resulting three-dimensional shape changes. Now, does it mean that the enzyme's shape must be changed if the order of the amino acid chains of the protein, which is held by the peptide bonds, are being changing? So, the three-dimensional shape must be changed of the enzyme, and for which the enzyme denatures. Hydrogen peroxide. Okay, so this is the example of the enzyme catalyst. What we understand about it is that. Um, the hydrogen peroxide, it is okay. Now in the diagram you can see it very clearly that the shape of the catalyst is specific to the substrate called hydrogen peroxide, while there's a DNA polymerase enzyme which is having the shape not similar to hydrogen peroxide. So there it's basically the concept that the enzyme must have the similar shape to the substrate to form enzyme substrate complex. If it is not same, so it will be not have any reaction. All right. An enzyme substrate complex forms when an enzyme and its substrate join together. The enzyme substrate complex is only formed temporarily. Okay, what we were saying before the enzyme catalyzes the reaction and the products are being released. So this is the temporary structure. Enzyme reactions can either be catabolic or anabolic. What does catabolic and anabolic means? It is basically the reactions of metabolism. Catabolic reactions involve the breakdown of complex molecules into simpler products, which happens when a single substrate is drawn into the active site and broken apart into two or more distinct molecules. Such as the examples are cellular respiration and hydrolysis reactions. Just wait a minute. Yes, the see that now this is the catabolic reaction, and in this catabolic reaction, the substrate enters the active site, and how the enzyme is been making two products from one substrate. So it means that it is breaking down the substrate, and this is called the catabolic reaction. All right. While in the anabolic reaction, it is vice versa, obviously, that the anabolic reaction involves the building of more complex molecules from one simpler once by drawing two or more substrates into the active site, forming bond between them and releasing a single product such as protein synthesis and photosynthesis. In the anabolic reaction you can also see this diagram. Two substrate molecules enter the enzyme's active site. All right, the two substrate molecules form a single product. So this is the example of the anabolic reaction. It occurs in protein synthesis and photosynthesis. Okay. Enzymes work by lowering the activation energy of a reaction, such as all chemical reactions are associated with energy changes. Okay. For a reaction to proceed, there must be enough activation energy. Activation energy is the amount of energy needed by the substrate to become just unstable enough for a reaction to occur and for products to be formed. Enzymes speed up chemical reaction because they influence the stability of bonds in the reaction. The destabilization of bonds in the substrate makes it more reactive. Enzymes work by lowering the activation energy of a reaction, and in doing so, they provide an alternative energy pathway. It means that the enzymes basically lower the activation energy. As you can see this diagram clearly, what is what's been happening that without the catalyst or without the enzymes, the activation energy was high. When the enzymes now bind the substrate and just react. So now it lower the activation energy. You must also be careful while see this diagram that from where till where the activation energy is and what does the activation energy means, because sometimes it appears in MCQs that state that whether A, B, C or D is the activation energy, and most people just complicate themselves as they're not seeing that from the substrate till the rising amount of the energy is called the activation energy, not from the substrate to the product or from the product to the energy rising. All right. How enzymes work? Okay, so in your O levels you have already learned this lock and key hypothesis, where the lock is the enzyme and the key is the substrate. This is what you have already learned. I'm also having a video on it. Yes, I'm also having a video on it of enzymes for the O levels biology 5090. You can also go and watch there. And what else is new here is that this means your shape as a wild active site of an enzyme is determined by the complex tertiary structure of a protein that makes up the enzyme and therefore is highly specific. Alright, so in the 1890s, the first model of enzyme activity was described by Emil Fischer. Okay, must not have to learn about this. Okay, so what he suggested that the both of the enzymes and substrates were rigid structures that log into. Okay, so in the lock and key hypothesis, there's a person ML which has already tell us that this is the rigid structures locked into each other. What are the rigid structures? The enzymes and the substrates, and are locked into each other very precisely, much like a key going into a lock. This is known as lock and key hypothesis. This was later modified and adapted to our current understanding of enzyme activity permitted by advance in techniques in the molecular sciences. Okay, now what is new is basically, and yeah, this is the diagram. You already know about this diagram. If you have not learned about this diagram or what is the lock and key hypothesis, this was the chapter I have briefly discussed in the O levels, and it is the same in the E level. But now here is the new thing, which is the induced fit hypothesis. What is basically the induced fit hypothesis? This is a bit tricky for you guys, as you have already learned from your childhood that the enzyme and substrate, and they are the lock and key, and they are the rigid structures combine and have a specific shape, but now we are also required to focus on their specificity. The modified model of an enzyme activity is known as induced fit hypothesis. What is this? Although it is very similar to the lock and key hypothesis, in this model, the enzyme and substrate interact with each other. What does it mean basically? The enzyme and its active site and sometimes the substrate can change shape slightly as the substrate molecule enters the enzyme. Enzymes and the active site of enzyme can change the shape slightly. This is called the induced fit hypothesis. In the lock and key hypothesis, this was simple that the substrate can be attached to the active site of the enzyme. But in this induced fit hypothesis, this is a theory where the enzyme can change their shape slightly so that can be bind with substrate. These changes in shape are known as conformational changes. Okay, this ensures an ideal binding arrangement between the enzyme and substrate is achieved. Okay, so I don't think so that this is what you're required to learn. That induced fit hypothesis is simply basically that the enzyme will be change the shape, and why it is changing the shape, because in the case uh, when the substrate is not having the specific shape to the enzyme, so then the enzyme and sometimes the substrate can also change the shape according to the enzyme's shape, so for which binding is very important for that, so that's why they'll be changing the shape. Okay, just give me a bit time, just wait a minute. Now here you can see in the diagram that the substrate does not fit perfectly into the active site. You see clearly that what are the specific shape, okay, in the stage 1. Okay, the active site changes shape, undergoes a conformational change to allow the substrate to bind enzyme substrate complex. Now this is called enzyme substrate complex. All right, at the fourth, the okay, at the third, the enzyme product complex. Now the products are being made, and at the fourth, the products are being released. Measuring enzyme activity. The progress of enzyme catalyzed reaction can be investigated by measuring the rate of formation of a product using catalase, measuring the rate of disappearance of a substrate using amylase, investigating catalase activity. In this investigation, the rate of product formation is used to measure the rate of an enzyme controlled reaction. Hydrogen peroxide is a common but toxic byproduct of metabolism. This means it must be broken down quickly. Catalase is an enzyme found in the cells of most organisms that break hydrogen peroxide down into water and oxygen. Hydrogen peroxide and catalase are combined with and the volume of oxygen generated is measured in a set time. The rate of reaction can be then calculated. Alright, so this is how the procedure will be happening. You're having the conical flask, hydrogen peroxide as a solution, and the potato cubes, source of catalase. Okay, it means that the potato must have the source of catalase. There is a tube transferring oxygen, and oxygen is collected in the measuring cylinder, and the water to exclude air during oxygen collection. Alright. This you must know, and as it is a chemical or sometimes your biological practical skills, so you must know what is happening right now and how to calculate and what are the basic apparatus with the readings. Investigating amylase activity. In this investigation, the rate of substrate disappearance disappearance is used to compare rates of reaction under different conditions. Okay, amylase is a digestive enzyme that hydrolyzes starch into maltose and glucose. Amylase functions best at pH 7 and 37 degrees Celsius. All enzymes operate best under specific conditions. This is obvious. Amylase and starch are combined, and this reaction mixture is then tested for starch at regular time intervals. This can be done by taking samples from the reaction mixture at each time interval and adding each sample to some iodine in potassium iodide solution. Starch forms a blue-black color with its solution. In this way, the time taken for starch to be broken down can be measured. The investigation can be repeated under a variety of conditions, for example, by altering pH or temperature, and the rate of reaction rates can be then compared. Alright, so there is a pipette and there is a tube which consists of amylase plus a starch solution, and then the mixture tested for starch are regular intervals. Yeah, you'll be taking 4 to 5 drops of amylase starch solution. You'll be taking from the pipette 2 ml as your practical. And spotting tile. Okay, so this is called the spotting tile, and each well contains 1 drop of iodine solution to test for starch. Okay, when you just add the concentration of the from the pipette of the amylase or starch solution into the spotting tile, so each boxes are you be seeing here. So this is not just a box, but this is like looks like a bit of the circle where there is a solution of iodine, and while you just dropping it, so the color will be changed from black, dark blue, black, and then being light and being light and then being light as if you move towards down. And through this, you can also measure that what is the highest concentrated and which is the lowest concentrated. Okay, colorimetry to measure enzyme activity. A colorimeter is able to measure Okay. A colorimeter is able to measure light absorbance, how much light is absorbed, or light transmission, how much light passes through a substance. Colorimetry can be used in any enzyme catalyzed reaction that involves color change. As the color breaks down, the transmission increases or light absorption decreases, and this can be used to measure the rate of reaction. For example, a colorimeter can be used to follow the progress of a starch amylase catalyzed reaction as amylase breaks the starch down into maltose. This can be carried out as followed. Okay, colorimeter calibration. This is an important step in a colorimeter investigation, and in this case, a weak iodine solution can be used to calibrate the colorimeter as the end point or 100% of transmission. Preparation of a star solution of known concentration, the stock solution, from which a range of concentration are made using serial dilutions method outlined in diagram below. What are the serial dilutions? So basically the serial dilutions are the dilutions you have been required to do in your A levels practical, and indeed in your AS you are required to just do only the serial dilution. There is also the simple dilution, but you are not required to do it. You are only required to do the serial dilutions. Following calibration and switching on the red filter to maximize the percentage transmission or absorbance. The colorimeter is used to measure the percentage absorbance or percentage transmission values. A calibration graph is then plotted to starch concentration excesses versus percentage absorbance of or percentage transmissions at y-axis. This is what you are required to do in your graphs of your practical. Okay, and that's the last thing, the serial starch dilution. One more thing you're required to understand is basically what does it means, and because many times that this is not only just a question but must be your practical also, and in your P3 of your biology 9700 of your AS level, you must perform it by having a test tube, and the solutions must be given to you, and there's I'll be discussing this in another video briefly in the detail about what is basically the AS level practicals required in the biochemistry and physics. So in the first step, test tube, so this is 1 centimeter cube, okay, and then it's 10 centimeter cube means 1% of a star solution. Okay, this is also called the stock solution. Why it is called stock solution? Because in the simple dilutions, we always take this as the stock. We always take the solution from the number one test tube, which is called the stock solution, which is the basic or the original solution from which we are required to prepare the other solutions. Okay, in the second test tube you can see that 9 centimeter and others are also called 9 centimeter of water be present in it, and there in the first test tube there was 10 centimeter water. All right, sorry, 10 centimeter 1% of star solution. Okay, and then we add 9% 9 centimeter cube of water, and then 0.1% star solution being added to it, obviously because it is 1 upon 10, so then we are required to multiply by it, or sorry, divide by it. In the second you'll be taking half and just halving the value of what you have been taking, but remember that in the third test tube where you'll be preparing 9 centimeter cube of water and 0.01% concentration. So this is how you take out the concentration, by taking the star solution from the test tube number 2, from where you have take the solution from the test tube number 1. Now just leave the test tube number 1 and just focus on the test tube number 2, from where you have take the star solution from the stock solution, and you have added the 9 centimeter cube of water, and now this is 0.1 percentage concentration, and then you have take again 9 centimeter cube of water and again take 1 centimeter cube of star solution, and this is 0.01, means now the value has been halving and then it's becoming lowest. So as you have been seeing that the 0.001 and then 0.0001 percentage concentration are the concentration is basically reducing. So in the serial dilutions, you are required to just uh, also half the concentration, which is reducing, and the serial star solution made using a dilution factor of 10. So each solution is 10 times less concentrated than the one before it, obviously. And in our one more video, I'll also try to perform it for you guys so that it will be very easy for you to understand. So till that is here, and I hope so that you understand what is basically a new thing for you from the O levels as you have been reaching to AS, and now what is new for you in your enzymes. I hope so that you will be scoring good marks. So till that it's here, and take care.